I'd like to invite to the stage our keynote speaker. I'm pleased to introduce Pinchas Cohen. He is the Dean of the USC Davis School of Gerontology, the Executive Director of the Esther Percy Andrus Gerontology Center, and the William and Sylvia Kugel Dean's Chair in Gerontology. His laboratory focuses on unraveling processes related to aging, diabetes, neurodegeneration, and cancer. And he is leading new initiatives, including the development of a Center for Digital Aging and the creation of tools for personalized aging. So please welcome Assay Cohen.
Thank you. So I want to talk to you today about a relatively new field in biology of aging, namely mitochondrial-derived peptides. And I'm grateful for the invitation from Audrey and others to be here with all my friends and students at the Buck. I should disclose that the work that I will present to you today, much of the technology has been licensed to a company that I co-founded and hold stock in, which is Cobar Inc., based in Menlo Park. And I should start by saying that my ultimate prescription for maximizing lifespan, health span, and brain span is diet and exercise. I think if we'll all follow that, we can lower healthcare costs to the trillions and increase lifespan and longevity and healthy aging. But the processes that occur related to diet and exercise clearly involve an organelle in the cell, the mitochondria, and I'd like to convince you today that much of the functions of the mitochondria involve the production of what we call mitochondrial-derived peptides, which have potent biological actions on aging-related diseases. So those of you who get the journal Cell, last night I got my electronic copy, and there was a wonderful review on the hallmarks of aging by a Spanish group, and they have increased the number of hallmarks of aging to 9. They're listed here. And of course, mitochondrial dysfunction is one of them. You're going to hear over the next day or two about many of the other ones, including senescence and so on. And clearly, aging is associated with multiple dysregulated, dysfunctional mitochondrial deficiencies, problems, and decline. And for those of you who are not experts in mitochondria, let me review some of the issues related to mitochondria. As you know, it's an organelle that was originally a prokaryotic cell that entered what is now the eukaryotic cell. We get our mitochondria from our mothers only, as opposed to the nuclear genome, which is equally shared from both parents. And the mitochondria's central function is respiration and energy production, but it also is in charge of apoptosis or cell death and the production of reactive oxygen species, which can be both an important metabolic function as well as a cause for processes that lead to aging and aging-related diseases. And there's a lot of evidence that mitochondrial function declines with age as a result of multiple processes such as mutations and damage to the mitochondrial DNA. But it's also important to remember that mitochondrial DNA haplogroups have been associated with longevity, in the case of Italian centenarians, with various types of type 2 diabetes, with neurodegeneration syndromes, and with malignancies. And you can see here, I think you can see my green little light here, that the mitochondria has a small chromosome, which is probably smaller than most nuclear genes, that was typically thought to encode 13 protein-coding genes, some transfer RNA genes, and 2 ribosomal RNAs that are unique to the mitochondria and operate with the bacterial coding system. And in spite of the fact that there's like maybe 2 handfuls of proteins that are made in the mitochondria, there are literally thousands of proteins found in the mitochondria, and they all come from the nucleus. They represent functional, structural, and signaling proteins. But how does the mitochondria talks back to the nucleus, to the rest of the cell, and to the body at large is still something that scientists are debating. And what I'd like to convince you today, if I can figure out how to transfer slides, is that mitochondrial peptides have a central role in that. So the first mitochondrial peptide that was identified was called Humanin. It was cloned simultaneously by 3 different groups. Nishimoto, who worked at Harvard and moved to Keio University, identified it as a survival protective factor for Alzheimer's processes using this death trap approach, where he took neuronal cells, added amyloid beta, and then transfected them with a library to see if he can protect from this amyloid beta-induced cell death. And the library came from the surviving region of an Alzheimer's disease brain shown here. You can see this is a brain donated by a graduate student. This is the brain of a full professor. And even though there are many areas in this Alzheimer's brain that clearly indicate that cells are dying, there are some areas that appear completely healthy, so he made a library from that and cloned out a gene which was very curious because it was actually a fragment of a cDNA, of the DNA of the mitochondria, which was expressing a peptide that induced protection. And at the same time, John Reed from the Burnham Institute cloned the same gene in the yeast-2 hybrid system as a partner of Bax that prevented cell death. And my group that was looking at it from a completely different area, looking for novel partners for a pro-diabetogenic molecule called IGF-BP3, identified Humanin as an IGF-BP3 binding partner and antagonist, published within a month of this Nature paper. And all 3 groups cloned the same mRNA and identified it to be identical to the mitochondrial 16S ribosomal RNA partial sequence. You can see here in our paper, we recapitulated the neuroprotective actions of Humanin on protecting from amyloid beta. So what is this Humanin molecule? So this is mitochondrial genome. There are many copies in each cell. Each one has this chromosome. And inside this chromosome, there's one region called 16S ribosomal RNA that is really a structural RNA that will form part of the ribosome of the mitochondria. And inside this region, there is an open reading frame, which is now named the Humanin gene, open reading frame. And what we and multiple other groups have then cloned are mRNAs that are encoded from this region in a separate transcriptional process where instead of the rRNA, an mRNA is made that contains this open reading frame. The open reading frame encodes a peptide, shown here. It's a 24 amino acid peptide, which is relatively well conserved in vertebrates all the way down really to zebrafish. And rodents actually have a little extension to distinguish in them. And this is a polyadenylated mRNA. It's translated not in the case of humans in mitochondria itself, but rather in the cytoplasm. And it's what I now call a gene within a gene. And we find this peptide using ELISA assays that we've developed against it in multiple tissues and fluids in the body. An interesting thing about how these things work, and let me say I'm going to describe to you today multiple mitochondrial-derived peptides from various open reading frames inside the mitochondrial DNA, and not all of them follow the same story here, but what we verified for Humanin in human cells, as opposed to other species, is that like all mitochondrial peptides, they are encoded in the mitochondrial genome, and we can eliminate Humanin production using a process of elimination of the mitochondrial DNA known as row 0. And the mRNA for humanin gets carried to the cytoplasm where it is translated in cytoplasmic ribosomes using the mammalian code rather than the bacterial code. And we've recently, in collaboration with the proteomic core at the Buck, have identified mRNA binding proteins that transport the humanin mRNA from the mitochondria to the cytoplasm. The peptide is then translated and gets secreted outside of the cell, as well as spending time in the cell to regulate biological functions. It's secreted through the standard process, and it has something called a signal-like peptide region that if you mutate it, it prevents the secretion. And then, as I'll show you later, there are membrane receptors that humanin and other MDPs can interact with. So it's a complex life cycle that's quite unique. I also want to point out that we have identified that even though humanin is a mitochondrial peptide that is secreted, humanin then goes back either from inside the cell or from outside the cell and targets itself to the mitochondria where much of its function, although not all of its function, is centered. So here you can see that these cells, which happen to be retinal epithelial cells, produce humanin, and that humanin, endogenous humanin, is localized in the mitochondria of these cells. If we take exogenous humanin and add it to cells, I can again demonstrate for you that it gets rapidly internalized and again gets targeted and localized to mitochondria. So it is a mitochondrial-encoded peptide that gets secreted out of cells to conduct multiple functions, like neuroprotection, as I've shown you, but also goes into cells and gets targeted to mitochondria. So in a series of papers from our lab and other labs, most recently, a couple of weeks ago, Sujang Kim and colleagues in my lab in OncoTarget demonstrated that there are 2 receptors for humanin, at least 2. One, which is the central one that we look at now, is a neurocytokine receptor composed of the GP130 receptor, and 2 other subunits. One is the CNTF receptor subunit and the WSX1 IL-27 subunit. We can inhibit this receptor in various ways and show that humanin signals through a JAK subunit in 3 different pathways involving the PI3 kinase AKT pathway, the STAT3 pathway shown here, and the ERK pathway shown here, where the kinetics of activation is different for each pathway. And in addition to this receptor that regulates cytoprotection, neuroprotection, and mitochondrial respiration, there's a second receptor that has been described called the FPR receptor, which humanin binds to and activates calcium flux and some anti-inflammatory processes. So there is membrane signaling, there's internalization, and there's mitochondrial targeting. And there's multiple biological effects of humanin, but I think it's important to show that it, among other things, one of the key effects is the regulation of mitochondrial function. For example, you can add humanin to cultured cells and show induction or more than doubling of oxygen consumption rate with seahorse, demonstrating increased mitochondrial activity. You can also show that the mitochondrial function, respiration, which is suppressed by various agents that are organic peroxides, can be protected by humanin, which also protects from other damaging effects of these kind of ROS-inducing agents. And finally, endogenous humanin, which we can downregulate or knock down using various siRNA approaches, and antisense approaches, you can see that relative to control siRNA in green, the addition of specific humanin siRNAs suppresses mitochondrial function and oxygen consumption. So the mitochondria is both the source, the localization target, and the endpoint effect of humanin in certain cases. Humanin also induces mitochondrial biogenesis, and you can see this here, both visually, that treatment with the same poison known as TBH, it's a hydrogen peroxide inducing agent, causes loss of mitochondria in this cell type, and co-treatment with humanin increases the number of mitochondria microscopically and also increases mitochondrial DNA copy number in these cells. So while I could focus on a lot of other effects of humanin, most importantly, protection from cellular toxins, which humanin has been shown to have, I think that my thinking today is that much, if not all, of humanin's activities involve protection of mitochondrial function. Now, we've learned a lot over the last 5 years or so about the physiology and the function of humanin in mammals through the development of an ELISA assay that potently and accurately measures humanin level in both humans and mice. And there are multiple papers we published on this, but we can identify humanin in plasma and CSF. And in fact, I haven't shown you in seminal plasma where it falls dramatically during aging. In fact, by age 40, the levels are 20% of what they are in a 20-year-old. The levels in circulation also fall to about 30 or 40% of young levels by the time you reach the ripe age of 80. Levels also fall in the brain, this is in mice, to less than half of young mice by the time the mice are middle-aged. As I'm going to show you in a minute, humanin is downregulated by the growth hormone IGF system, which is known to be progerotic or to decrease lifespan. It is related to insulin sensitivity, obesity, diet, and exercise, and I'm not going to review the details of that. It is low in Alzheimer's disease patients' CSF, and I'm also not going to be able to review our data on that for you today. And as you can see here, it's directly related to mitochondrial DNA copy number in in patients, and this is humanin levels in plasma are higher in patients with high mitochondrial DNA copy number in white cells as opposed to low mitochondrial DNA copy number. In particular, we published a couple of years ago that in patients who are being evaluated in the Mayo Clinic for endothelial dysfunction using cardiac catheterization and comparing patients with normal endothelial function to patients with poor endothelial function, but no clinical signs or symptoms of heart disease. So you actually need to do the catheterization to identify the mitochondrial, the endothelial dysfunction, have humanin levels that are less than half of the healthy controls. And to complement that, we conducted a study in a strain of mice, which is the ApoE knockout mice, which is a model for atherosclerosis if you feed it a high-fat diet. And you can see that untreated, these mice develop these nasty plaques on their arteries. And humanin treatment in the form of the humanin analog HNG, which was given daily for 3 months, practically eliminates the plaque completely, about 90% of it. This was shown in a separate publication, demonstrating that the broad physiological effect of treatment with this peptide are potent disease preventing. And I told you that there's a connection to growth hormone and IGF. Growth hormone transgenic mice, which we obtained in collaboration with Andy Bartke, which are large, they're short-lived relative to normal mice, and they develop various diseases of aging, have dramatically reduced humanin levels, while the growth hormone deficient low IGF mice, known as the Ames mice, which have no growth hormone, they're much smaller, and they live substantially longer, have elevated humanin levels. And in fact, in both mice and children treated with growth hormone or IGF, we can demonstrate that both agents lead to suppression of endogenous humanin. So there is a yin-yang relationship between growth hormone and IGF, compatible with the presumed longevity-enhancing roles of this mitochondrial peptide, which oppose or are related to the geronic effects of the growth hormone IGF system. So to further evaluate the potential relationship between humanin and longevity, Kelvin Yen at USC, my colleague, developed a humanin transgenic worm and has demonstrated that this worm has a pretty substantial increase in lifespan. And to find a human correlate, we corroborated with Nir Barzilai, whose mugshot was shown earlier this afternoon, and looked at a cohort of centenarian children. And Nir Barzilai always told me that the problem of working with centenarians is that the control group died 30 years ago. So instead, he has the children of centenarians with age and sex match controls, and you can see that centenarian families have humanin levels that are about 3 times higher. Again, linking humanin to human longevity. So one of the biological effects of humanin, apart from the disease prevention at the atherosclerosis level, is the metabolic stabilization, what I call metaboloprotective effects. And we've shown this in a series of papers that humanin improves blood sugar levels in Zucker diabetic rats in acute treatment. It prevents diabetes in both type 1 and type 2 models. And it acts centrally by targeting the hypothalamus and controlling the hypothalamic hepatic pathway, which is mostly vagal neurological in nature, by acting on STAT3 activation in the hypothalamus. And that's interesting because even though it's given systemically, we can specifically block STAT3 in the hypothalamus using ICV injection of an inhibitor and show that we lose the systemic humanin effect. We can also get a much more potent effect of small amounts of humanin if we give them ICV directly into the brain, so it targets the hypothalamus as opposed to systemically, even though the systemic effects are quite potent. And I think it's interesting that a lot of people talk about a connection between Alzheimer's disease and diabetes. People have even called Alzheimer's type 3 diabetes, and humanin targets biological processes in both conditions. Now, about 4 or 5 years ago, we were thinking about the concept that there's an open reading frame here in this mitochondrial chromosome, and it has the ability to encode a peptide with potent and exciting biological activities. And we said, are there additional open reading frames? And we initially limited our quest just in the small region node of the 16S, and we identified 6 additional peptides, which at the time we called small humanin-like peptides, or SHLEPs. And in some crowds that get a laugh. But there are various sizes of peptides. We synthesized all of them and developed antibodies against all of them and demonstrated that they are indeed a group, I don't know if you can call them a family, a group of naturally occurring peptides that regulate apoptosis, insulin sensitivity, and inflammatory markers. This was published in April in the journal Aging. And I'm only going to show you a couple of highlights from our current analysis of these 6 peptides. The ones that we're most interested in at the moment are SHLEP2, which has potent metaboloprotective, if you want, anti-diabetic effects, but also a very, this lost a little bit of the lines here, but a very potent protective effect on neuronal cells against amyloid beta, more so than Humanin. We believe it may have a potential role in both Alzheimer's therapy and diabetes. But we were quite surprised that a second SHLEP, we call SHLEP6, in fact, unlike the other mitochondrial peptides, which appear to be protective, can induce cell death in cancer models. And we've now looked at this in many other models, in vitro and in vivo. And SHLEP6 is a pro-apoptotic potential cancer therapeutic that we're exploring, as well as other peptides of this family. I've told you about the endothelial protective effects of Humanin, which were published in another paper here, showing that the addition of oxidized LDL induces reactive oxygen species production, and this is suppressed by Humanin, but not by a scrambled peptide. It is also equally suppressed by SHLEP2, which suggests that these mitochondrial peptides synergize and perhaps are coordinated in their activity as protective agents. So one area that always concerned me about this protective peptide therapy for chronic diseases of aging is that what if it also protects cancer cells, and cancer is yet another disease of aging that's quite prevalent, maybe would be protecting against diabetes and Alzheimer's, but enabling cancer. So to look at that, we started a series of trials in mice, published in now 3 papers, where we looked, first of all, at the ability of Humanin and other mitochondrial peptides to serve as adjuvant therapy in cancer to protect against chemotherapy side effects. So in this case, you can see both germ cells death. This is a side effect of chemotherapy that causes infertility in both males and females. This is male mice. You can see that there's germ cell death that's induced by chemotherapy and is protected by Humanin, but also the white cell decline, the leukopenia that occurs in response to chemotherapy, cyclophosphamide in this case, is protected from by Humanin. These are in mice that don't have a tumor, they just received the chemotherapy. So when you do this in mice that actually are tumor-bearing, in this case, this is a melanoma, and you look at the ability of this melanoma to metastasize, to our delight and surprise, you can see that, in fact, Humanin is synergistic with chemotherapy in reducing the growth of these tumors and inhibiting the development of metastases. And let me say that this is very similar to the effects of caloric restriction and fasting that Walter Longo at USC has demonstrated. And coupled with the fact that Humanin itself rises in response to fasting, we believe that Humanin injection act essentially as a caloric restriction mimetic therapy. And in a review that I wrote a couple of years ago in JNCI, I proposed that Humanin and perhaps other mitochondrial peptides may serve as both synergizers, adjuvant therapies on inhibited tumor growth, while at the same time protecting from chemotherapy side effects. And again, this is the same type of evidence that have been demonstrated for dietary interventions, but that in those cases, or for those people who are unable or unwilling to take the dietary approach, this kind of pharmaceutical approach holds promise. Now, we've recently completed a very extensive study of humanin in aging, where we looked at middle-aged mice that are 18 months old and treated them with twice-a-week injections of humanin until the age of 32 months, which is quite at the end of life. And we wanted to see what happens to these mice, hoping, first of all, that we might improve their lifespan. And let me start by saying that mean life expectancy or lifespan of these mice was not changed, in my opinion, probably because we were underdosing them dramatically. The half-life of humanin is about 30 minutes, and we were giving these twice a week. Nevertheless, we showed dramatic improvements in health span and brain span. And you can see here that this low dose of humanin did result in some reduction in body weight at some point, but not at the very last few months of life, without change in food intake. This was also associated with improved body composition, higher lean body mass, and some reduced fat mass. There was also evidence of lower blood sugar, lower IGF-1, again, a caloric restriction mimetic effect, and improved insulin sensitivity measured by increased BP1 levels. There was also dramatic inhibition of inflammatory cytokines, in the case IL-6 and TNF-alpha. And importantly, what we call brain span or neurocognitive function was improved in tests such as the Barnes-Maze test, the Y-Maze, as well as this neuromuscular test known as Rotarod, showing that just twice-a-week injection of this very short-acting peptide can improve all of these functions, as well as lowering insulin, which together I call caloric restriction mimetic activity. And in the same study, we were quite excited to see that the normal process of cardiac fibrosis, which is an integral part of the aging process, but is also accelerated in a number of other conditions. And you can see, compared to young mice, old mice have very enhanced cardiac fibrosis, but the old mice that were treated with humanin have essentially a rejuvenation of the heart muscle. There's a prevention of the development of cardiac fibrosis. And we're very excited about this observation that was, again, done in collaboration with the Lerman Group at the Mayo Clinic. Now, switching gear, for example, for a minute, one of the areas that I'm very interested in is minority aging and health disparities during aging and aging-related diseases. And we were interested in looking at the mitochondrial genome in African Americans and Hispanic Americans. And you should know that there are numerous diseases that have mitochondrial DNA-related sites that can cause cancer, diabetes, and heart disease, which are uniquely present in people with African ancestry. So we wanted to just take that approach and empirically measure whether humanin levels are different between ethnic groups. And you can see here that African Americans, both male and female, have lower levels of humanin and lower different distribution of humanin levels. And the lower humanin levels may be facilitatory to some of the aging-related diseases that African Americans are afflicted with, which lower and shorten their lifespan and increase their rate of various diseases, particularly Alzheimer's, diabetes, and heart disease. And in fact, we were intrigued when we started sequencing mitochondrial DNA and noted that there is a previously described polymorphism known as RS2854128, which is right at the site of humanin in the 16th region. It's previously not been associated with any kind of pathology or phenotype, and we measured humanin levels in both the reference and the alternative form of this polymorphism. And in fact, people with the alternative form of this polymorphism have lower humanin levels, again demonstrating that mitochondrial DNA, the origin of humanin, is through what must be regulatory transcriptional processes that this SNP might be modulating affect humanin levels. And finally, I'd like to switch gear to tell you about yet one more mitochondrial peptide that we're particularly excited about, which we call MOTS-c, or MOTS-c, depending how you want to pronounce it. But MOTS-c is a peptide that's encoded from a region in the mitochondrial DNA known as 12S, right next to the 16S. It's, again, a structural RNA, and it's a gene within a gene. And as you might guess, MOTS-c means that there's also a MOTS-a and a MOTS-b, whose function my colleague David Lee is unraveling. And MOTS-c is another gene within a gene in the 12S region, encoding a 16 amino acid peptide. It's even smaller. This was published last year in Cell Metabolism. And again, like humanin, the levels of MOTS-c fall, depending on the tissue, fairly dramatically with age. And MOTS-c improves glucose homeostasis in mice, and it does so in what we'd like to think of as an exercise mimetic, as opposed to a dietary mimetic. And here is an experiment where you compare young to old muscle that's been removed from mice and ex vivo incubated with insulin, and you can see that glucose transport, a measure of insulin action, which is known to be impaired with aging, goes down with age. But if you take old mice, treat them for MOTS-c for 1 week, and then take the muscle out, there is essentially restoration of the insulin sensitivity in these older mice. This is something that exercise of mice, old mice, also does, but just fasting does not mimic. So we think MOTS-c is an exercise mimetic. We demonstrated that the main effects of this peptide in vivo using what's known as the euglycemic clamp technique, which is the gold standard for assessing insulin action, improves the insulin action glucose utilization rate primarily on muscle with no real change in the liver, where, for example, anti-diabetic drugs like metformin have their main action. And we've demonstrated that the biological effects of MOTS-c involve first induction of intracellular ACAR, which leads to the activation of AMPK phosphorylation, and then to the activation of multiple downstream insulin-dependent signaling pathways that facilitate normalization of glucose transport and other insulin actions. Again, both similar to exercise mimetic activities, of course, of natural exercise, as well as some drug of abuse like ACAR. It is the Olympics, and they haven't developed a MOTS-c assay for cheaters yet, but I'm sure that's coming. The most exciting effect, I think, of MOTS-c is shown here, and it's a 2-month experiment looking at the effect of high-fat diet, which compared to normal mice eating a low-fat diet, which hardly gain much weight, and if they're also treated with MOTS-c, there's really no significant effects on weight. When they eat the high-fat diet, they, of course, have pretty dramatic weight gain, but the high-fat diet plus daily injections of MOTS-c shows a dramatic reduction in weight gain. And in fact, in data not shown here, you can take obese mice and treat it with MOTS-c and see weight loss. This happens without change in food intake, but in metabolic changes, you can see that activity and HIT production go up. And more importantly, or equally importantly perhaps, the development of fatty liver, or hepatic steatosis, which is now considered to be one of the main precursors to a lot of metabolic problems, including diabetes, is dramatically affected and normalized in mice treated with MOTS-c. And these are mice that don't actually develop diabetes yet. They're more like pre-diabetic, but they're cured of their fatty liver. Their insulin sensitivity is restored. And this is, even though this is not an aging model, it's certainly a normalization of metabolic dysfunction. And in now probably 19 reviews on muscle and diabetes metabolism that have looked at MOTS-c and other agents, I think the summary that I'd like to give you on MOTS-c activity is that it's a mitochondrially encoded peptide that is secreted and then goes back to cells and acts to activates ACAR, AMPK phosphorylation, leads to insulin sensitivity normalization and weight loss, acting both on muscle and on fat. And we were particularly intrigued to see a follow-up paper to our work coming from a Japanese group that studies centenarians in Japan, that has identified a MOTS-c related mutation, and that is exclusively seen in subjects with exceptional longevity in Japan, where there is a modification of MOTS-c sequence that seems to be associated with exceptional longevity. And we're now studying how this peptide is uniquely able to mediate some of the effect of MOTS-c at lower concentrations. So I'll finish off by saying that I've mentioned today about 8 peptides that are encoded from the mitochondria, but in fact there are hundreds of potential regulatory peptides encoded from the mitochondria that we believe play important physiological roles. We're studying many of them. We think they might be related to a host of diseases, and many of these are in various stages of evaluation in our labs. And I've shared some of them with you, but in fact it's a novel and growing group of hormones, and I think they do deserve the title hormone. And they have both therapeutic and diagnostic potential. And for the scientists in the audience, we've created a comprehensive set of tools, reagents, assays, antibodies, and transgenic mice, some of which I hadn't had an opportunity to talk about. And we'd love to create new collaborations. And I think that I started by saying diet and exercise is the key to health, and that the mitochondria is the key component of making that work. So I'm grateful for having the opportunity to speak to you and to the SENS Foundation, and of course to all of the brilliant, wonderful people in my lab, Kelvin Yen, Sujang Kim, and many others. Some of them are here in front of the mitochondria that is on the ground floor of our lab, and many, many collaborators from Amir Lerman, David Lee, Nir Barzilai, Walter Longo, and most recently Judy Campisi here at the lab and her students. So thank you.